So we're going to talk about this obscure little rabbit that occurs here in Maryland. And I want to start with a confession and an apology. I am not a rabbit expert per se. I work with small mammals. My Ph.D., a lot of my research background is actually in the tropics, working in places like Brazil and Panama and Venezuela. I have done a little bit of rabbit work, which is why I'm here, and we will talk about that at the end. I am a mammologist. I do study mammals. I do teach about these mammals. And as for apologies, when we scheduled this was a while ago, and I guess I wasn't really thinking about this being the second week of classes at the university and how that might wear on me, and that I didn't even know that Thursdays would be my long days, long field days with lab classes. So hopefully I can be my energetic, effervescent self here, but if I fade a little bit or forget what I'm saying, you will have to forgive me. So this is the critter we're talking about, the Appalachian Cottontail, Sylvilagus obscurus. I should have a pointer. That's a pointer. You can see its range here through the Appalachian Mountains, so up into Pennsylvania ways, down south. Comparing it to the Eastern Cottontail, the biggest thing is it's smaller, only getting up to about 2.5 pounds, whereas Eastern Cottontails can get as large as 4 pounds. Here in Maryland, it's restricted, as far as we know, to areas of Allegheny and Garrett County, and we'll talk about some of the current sort of range data on them toward the end, and sort of the search for them, if you will. According to the IUCN, it's near threatened, which means there are credible threats to their populations that could put them at risk of becoming threatened and then endangered. In Maryland, it's considered S1, so in need of conservation, and it is technically considered a non-game species. Now, as we'll talk about here in just a little bit, you can't really tell it from the Eastern Cottontail, which is a popular game species. So it is still harvested. It's just not considered a harvested species. So what is it? Well, it's in the order Lagomorphia. So this is the order that contains rabbits, hares, and pica. As an order, there's two really distinct characteristics of it. The first are these fenestrated skulls that you see here, this sort of lattice work structure on the skull. And that's an adaptation to that hopping, to that ricochettal locomotion. It's basically a shock absorber built into their skull. The other morphological feature of them is they have these big buck teeth incisors that you're familiar with, right? The big Bugs Bunny teeth. Very similar to rodents, big, large, ever-growing incisors. But then they also have these two little peg-like incisors behind there, which differentiates them from rodents. And there was a long-standing debate. These are sort of superficially rodent-like with those big teeth. But there was a long-standing debate, are they really close or not? Some people arguing, well, yeah, they're really close to rodents. Other people saying, well, no, it's just sort of convergent evolution. And if we look across mammals, we do see a lot of organisms that evolve those sort of two front teeth. I'm going to go back up, hopefully. The two front teeth, this space, which we term a diastema, and then the rear grinding teeth. And we see it in lots of things. Really, if you think about the teeth on a horse or even a deer, very similar without the ever-growing incisors. But we see it in species we know aren't very close, like the wombat here. But you also see it, this is an eye, which is a primate, has very similar dentition. In fact, this eyes were classified as rodents for a while. Now, long-standing debate over how close rodents and lagomorphs really were. That's recently been settled with molecular work. So we get what we call the grand order Gleries here, which is Rodentia and lagomorphs. And I like to mention this or bring this up for us is so rabbits and squirrels and mice and rats are your closest living relatives in North America. Here we are in the primates. The primates are part of a group called the Euaconta Gleries with the Gleries and also flying lemurs and tree shrews. But so they're our closest living relative. And despite the fact that lots of other things evolved it, both of these guys evolved it. While we're on the subject, another thing I often like to point out, there are things that some of us assume everyone knows. And so because we assume that, we never tell anybody. And then nobody really knows it. So what is the difference between a rabbit and a hare? Rabbit, hare. Rabbits tend to have shorter ears, and shorter legs. Their young are born helpless, blind, hairless, and as a result, they build burrows and nests. If you've ever kicked around in a field and kicked up a nest of sort of a ball of fur, it's often a rabbit's nest, right? Versus a hare where we're going to have long ears and long legs and very precocial young. So young are really able to run and move right away. So we don't get any nest building in the hares. Now, in Maryland, there was, maybe is, but at least used to be one hare species, the snowshoe hare. Even when it was here, it seems like it must have been pretty rare. From my searches, I could come up with 10 total records in the state. And this would have been, a species that was harvested, trapped, hunted back in the day. So unlike the Appalachian Cottontail, which we'll get to, there should be some records of it, and we don't really have them. The last confirmed was in Garrett County in 1957, officially declared extirpated in 1986. Some of the sources I looked at say, well, there's populations in Virginia and Pennsylvania, so there may be some kicking around if you got into the remote enough regions of Garrett County. If you found one, people would be interested. But what we're here to talk about today are rabbits, right? And something really very few people think about is how many species of rabbit there actually are. We have the Eastern Cottontail, which is your typical rabbit. It's a rabbit you're probably most familiar with. Even those of you who might live out here in the Appalachian Mountains, if you see a rabbit, you're probably seeing an Eastern Cottontail. very common, very widespread. But then throughout its range, there's all these other species, right? The Appalachian cottontail we're going to talk about here today, marsh rabbits and swamp rabbits and New England cottontails. Then we get out in the mountains and there's all kinds of species. Then as we move through Central and South America, we get species. Uh, I use this map because it fits the screen a little better and we're not really concerned with Central or South America. There's a whole other group of species in South America. And really, with all of these, we have this situation where uh, people, before we started the talk, I mentioned sort of the difference between the New England and the Appalachian, where the only way to tell them apart is either know where you are or look at the genetics on them. And the same holds for a lot of these. Many of these species out here I know basically nothing about, but swamp rabbits and marsh rabbits. If my mammalogy class is getting on my nerves, I'll bring these out and tell them they have to know them. So I'll have out New England's, Appalachians, marsh, swamp, and easterns, and tell them they have to tell them apart. But really, marsh rabbit's a little bigger than some of them on there. But really, unless I told you where it was, you wouldn't be able to tell them apart. So if we look here, so here's the Eastern Cottontails range, all through that area. And then, as you saw in the previous slide, these numerous little bits in there. So, as was asked before, how do we tell an Eastern from an Appalachian? Well, we did mention the size. The Appalachian is a little smaller than the Eastern. Lost my button. The Eastern should have, if you read the field guides, a white spot on its head, whereas the Appalachian has a black spot between its ears. So there is a difference there. The Appalachian has shorter ears that tend to be lined with black. So there are some color differences between them. But as one site I read said, they didn't read the field guides, so they don't know what they're supposed to have. And not all of them not all of them have it. Sometimes you can take a guess at what you're looking at based on the habitat. If you're in sort of thickets, excuse me, rhododendron thickets, shale areas out here in the mountains, higher chance you're seeing an Appalachian. But really, other than genetics, which we'll talk about at the end, the way to tell them apart is based on this suture here at the nasals, where the nasals match the frontal, right? So you see here, these are the nasal bones. There's the nose of the animal. It is where that joins the frontal here. On the Eastern, you can see how this line is really smooth, right? It's a nice smooth line. Whereas here on the Appalachian, it's a jagged line. It's a crooked, jagged line. And that is distinctive. I mentioned gestalt earlier. You can stare at them sometimes and convince yourself you're looking at one or the other, but generally, if you look at it, your first instinct, once you know what you're looking at, is right. This is a fairly distinctive jagged line, and this is a fairly distinctive smooth line. So we can tell them apart relatively easily if we have a skull in hand. We don't need any molecular. Where did the Appalachian Cottontail come from? It has a very short history. It wasn't recognized until 1992. Up until then, it was considered the same species as the New England cottontail. Morphologically, the Appalachian cottontail and the New England cottontail are indistinguishable. You cannot tell them apart. They do differ in chromosome number and then some skull measurement. So you would really have to measure a bunch of them. Fortunately, or for whatever reason, or fortunately, if you're south and west of the Hudson River, you have Appalachian. If you are east and north of the Hudson River, it would be a New England. No, I thought that's okay. So, what do we know about the Appalachian cottontail in terms of its natural history? It occurs at higher elevations, as you would expect, 2,000 feet or more. Like rabbits in general, it's crepuscular, sort of dawn and dusk activity patterns. It And this was sort of meant as a joke, but it is also true. It breeds like rabbits. It'll breed throughout the warmer months. Its reproduction is probably tied to food availability, which is fairly typical of rodents and lagomorphs and really small mammals in general. If they have enough food, they will breed and have litters. Um, Generally, a female will have 3 or more litters a year, with each litter having 5 or more young. So, prolific breeders. They probably don't live much longer than a year. All the data would suggest that. They do like dense cover areas, mountain laurels, rhododendrons. There's been some radio tracking studies done, and they tend to stay in cover. So they don't like to get out into the open areas as much as the Eastern Cottontails tend to. And I tend to think of them, and this is true of a lot of animals, kind of the difference being escape methodology. The Eastern being a little larger, little stronger, little faster, maybe more out in those open areas and using straight line speed to escape predators, where the Appalachian, a little smaller, a little easier to move around up underneath some of these dense cover areas, get in there to escape. And then diet, barks, twigs, and leaves. They do eat a lot of tree seedlings, black cherry, oak, things like that. Spring and summer, they tend to eat more grasses, which obviously, when the grasses are available, that's a higher quality diet than chewing on bark. In terms of threats, we said they were near threatened, which means there are reasons to keep an eye on it anyway. Biggest ones being habitat loss and habitat change, especially if you consider that use of habitat where they like sort of that dense cover and tend to avoid those open areas. As we convert areas to farmland, to grazing land, to those kinds of open areas, that can be a problem for them. There is a disease that is spreading called rabbit hemorrhagic disease, which you see here, very prevalent in the Southwest. There are cases in domestic rabbits on the East Coast. As of yet, no cases in wild rabbits, but it can be fatal for rabbits, so it is something being monitored. Increasing deer populations results in some of that habitat change, um, and then competition for uh, food. And I put hybridization with the Eastern in parentheses. I don't have an answer there for you. I read one source, a non peer reviewed source, but one source that was talking about threats to the Appalachian cottontail that listed hybridization with Easterns as a threat. Now, they differ in chromosome number. So if they are hybridizing, they aren't producing viable young, which is actually would be bad for their populations, obviously. Now, what I could not find was any peer-reviewed documentation of that. So, and I asked Dr. Seton, who we'll talk about here at the end, that's doing some of the genetic work on these guys now, and he has looked for it too and can't find documented cases of it. But it was mentioned by at least a credible source as a threat. So I figured it's worth mentioning, but I put it in parentheses. So now, why we may not be as SMRT as we think we are. I didn't have a good visual for this, so I went with Homer. We sometimes think we're really smart when we're really much more not as smart. So obviously, the Appalachian cottontail is very difficult to study. If I can't reliably see it and point to it and say that's an Appalachian cottontail, most studies I need to collect individuals, right? So I have to go out, physically capture them, kill them, clean them, get their skulls to determine, do I have an Appalachian or do I have the Eastern? This can generally limit your numbers. Obviously, when we do scientific work, there are a lot of restrictions if we're going to collect on how many we can collect on there. Studies have tried, or studies have utilized hunter kills, so asking hunters to turn in usually the head, which the hunter doesn't want to keep. On that, you get limited participation in those studies, and hunters tend to hunt open areas. So you may be missing Appalachians that are there. All of this makes it very expensive and time consuming. Even if I don't kill individuals and collect them that way, if I have to capture them to get DNA, it's very expensive, time consuming. And this results in many of the studies having a confirmation bias. These things are difficult and hard to study. So if I want to go out, if I'm charged by, the local agency, the DNR or Fish and Game or whoever, to survey the Appalachian cottontail, I'm going to tend to go to places where I think it is, right? So then we end up confirming the habitat associations we think it has in there, which is how a lot of the studies have gone. And so if you really think about what I told you earlier about its natural history and its use of habitat, it does rabbit-like stuff in mountain-like habitat, which we kind of know, right? It's a rabbit in the mountains. So it breeds like a rabbit. That was the joke. Eats rabbit food, right? And it's in habitat, mountain laurel, rhododendron, shale areas that you find in mountains. Now, why our ignorance might matter. This is a species in need of conservation. This is a managed species in the state, and really throughout its range, it's being managed, right? Not really understanding where it is and what its habitat use might be can um, result in us misplacing conservation areas, having conservation areas that are too small because we say, well, it only uses this little bit, this type of habitat. So we only need to conserve this little bit of area, and the rest of that we can put to other uses. Also, if it is much more abundant than we think, we could be directing conservation money, limited amounts, increasingly limited amounts of conservation money to working on it that could be spent elsewhere. If we don't fully understand its use of habitat, its diet, things of that nature, our management plans can be far too narrow. And I mentioned earlier, I said some radio tracking studies showed that they never left the cover. But again, that could be true that they spend all their time in that dense cover, or it could be an artifact of the radio tracking studies that when people are out there tromping around with radio antennas to locate them, they don't venture out into the more open areas, right? When they detect a predator. And when you have limited samples, because the species is rare, you're working where you think you can find it. You have, I believe that study had less than a dozen individuals tracked. You can get some of these, well, they don't use open areas when in fact it's an artifact of us not having a great measure of where it is. And the biggest one, probably the most important reason our ignorance is important, is we could be missing trends in population size. We just kind of assume it's rare and we don't really know what's going on. So this is actually obviously not the Appalachian Cottontail. These are legomorph studies from the Western US. This is one that is looking at actual surveys. So going out and counting how many legomorphs total. So this is combining all the species that were out there. We're seeing, and you see sort of this big drop off in this one study. And if I would have pulled out some of the other graphs from this study, if you looked at hunting numbers and hunter harvest and things like that, all of them would show this massive decline. But if we really look at this with numbers, they are staying fairly steady, except for coming off this weird peak from the mid 60s. So it's really important in management and conservation to have a good idea what's going on with the species. Something the Appalachian Cottontail doesn't seem to want us to have. So just a couple of examples of species that are the opposite of the Appalachian Cottontail. Porcupines in the state of Maryland. You can see here on the Porcupine Range, we do get a little red dot out there, out here, and a couple of dots south. Porcupines in this state declined fairly dramatically to the point of maybe being extirpated, and have since rebounded. And the range of the porcupine in general is expanding, which is a really interesting range expansion because the porcupine is a northern species. With climate change, we generally expect northern species to keep going north and southern species to come up to replace them. But we have the porcupine rebounding in populations where it's now quite abundant out here in Allegheny and Garrett counties, quite frequently encountered, and seems to be expanding its range, especially into some of these areas here down in Virginia. But with the porcupine, right, I know that has been the trend in the population because this is an obvious unmistakable animal. People know when they see it. Another example I had to throw in because I just think this is amazing. Armadillos. So here on the right, each of these dots represent confirmed armadillo sightings. And you can see there are a couple up here in Ohio, some northern, all the way northern Indiana border with Michigan, some around Chicago. We know there are breeding populations as far north as Indianapolis here. And if you look at the potential range, right, I kind of cut off the eastern part of the state. I'm sorry, the eastern part of Maryland is on there as well, but all the way up well past us. But again, with armadillos, unmistakable. No one sees an armadillo and mistakes it for another species. And part of the reason I find this really amazing, here we see the range in 95. I interviewed at the Illinois Natural History Survey in 2004. So right after I completed my Ph.D. At that time in 2004, so 21 years ago, there was the first confirmed record of an armadillo in Illinois. And I thought that was pretty amazing that they found an armadillo way, way down here. Now we have breeding populations all the way up here into Indiana, southern Illinois, on there. So we have this problem with the Appalachian Cottontail that it's really hard to study. And because it's really hard to study, we sort of lack the ability to just go out and go to all the different habitats out there and go down farther east in the state to lower elevations and look for it and just see everywhere it might be. But there is a way we can get around this. We can amplify DNA from scat. So this is generic little diagram of polymerase chain reaction, PCR. If you've watched any police show ever, they use PCR, right? So we take the DNA, we have little primers, right? The primer gets the DNA replication going, and we just make copy after copy after copy of the DNA. So we can take a little bit of DNA and get a lot of DNA, right? When the animal defecates, every animal sheds cells. So the sort of outside of the scat has cells attached from which we can get the DNA. We amplify that DNA, and if we use primers specific to the species we're looking for, we can identify the species, right? So now we don't have to kill rabbits or even capture rabbits. We just can go out, gather scat, right? And gather the scat from all kinds of habitat, all kinds of areas, and see what we have. So, wider areas. So, this is a study that got me invited here today. A former graduate student, Kelly Russo, she went out and she sampled 10 historic sites that Appalachian cottontails were known to occur and 30 sites where they could potentially occur based on the assumed habitat associations. Now, again, you notice we still have that bias built in there, right? We're going to places we think they are. This was really the sort of earliest attempt at trying this, so wanted to be successful. It also does involve having to go out within, I believe the protocols say 3 days of a recent snowfall when the temperature is not above a certain, hasn't increased above a certain level or decreased below a certain level. And then you have to walk the areas and find rabbit tracks and then collect the scats and bring them back. So it is still fairly labor intensive. But she was able to amplify the DNA and tell the two species apart based on um, highest peaks. She actually sequenced the DNA to do this, which is an extra step we've actually gotten rid of. But here's her results. So I believe there were 7 sites where no scat was found. But there were sites with both Eastern and Appalachians, sites with only Appalachians and sites with only Easterns. The highlighted sites are the historic sites. So you see 3 of the historic sites had both, with only one actually having just the Appalachian, right? But what this shows is the species is far more widespread than we thought, right? So, right, founded 4 of the 10 historic sites and founded 18 new sites, right? And co-occurring with Easterns at 10 sites, right? But this raises a question. So this shows the species being quite a bit more widespread and abundant than we thought. But the big question is, well, what's going on? Has it always been abundant? Has there always been lots of them running around and we were just really bad at detecting them, right? Which is possible. Or, right, are populations actually increasing in abundance, right? We mentioned the spread of, clearing areas for agriculture, clearing lands for pasture, favoring the Eastern Cottontail, right? But some logging operations create dense, scrubby cover near the ground. Overgrazing by deer, we said deer increasing in population can be a threat, but deer graze on a lot of things, but they don't really like mountain laurel. They don't really like rhododendron, right? So they actually can lead to increases in that sort of scrubby habitat they like. So maybe the Appalachian cottontail is doing better than we think, or maybe they go through population cycles, being occasionally very abundant and then declining. This is very common for lots of small mammal species. A lot of the places I worked in the tropics, you could go there one year and you would catch lots of everything. You would say, wow, this is an amazingly diverse forest full of all these different, that would be rats, mice, and little possum species. And then somebody would go back 3 or 4 years later and they would catch nothing. We do see these big booms and busts in them. So maybe Kelly just happened to hit a high in the population cycle. And if we go do this again, we're not going to find them. So what do we have to do? We go do it again. So Dr. Bill Seton here at Frostburg State is working with the Maryland DNR. He's doing a slightly different molecular technique with the scats where he's not sequencing, he's just running through a gel. I asked for and did not receive a photo of one of their actual gels. So this is just the idea. Smaller pieces of DNA can move faster through the gel than larger pieces. So you can get different banding patterns to separate out the species. Right now, We have some roadkill rabbits, so I can pull the skulls, look at the skulls. We can be sure of what it is, and then you sort of ground truthing the technique. But the DNR is out collecting scats all the way west to Frederick, so over wider areas. Um, there's still some potential for bias. DNR is really collecting scats in habitat we would think they're in, right? But it's better, at least we're hitting a wider area. We're increasing sample size. And if this works, I'm going to try to convince Bill that we need to go out and do something closer to systematic sampling. Um, and get scats through wider areas, but still making sure this technique works. So, what can you do for rabbits? I like the word rabbitat, so I had to use it. Um, if you live out here, especially in the western part of the state, right, encourage rabbit habitat. Probably Easterns, almost certainly Easterns, but I am out here in Garrett County. When I first moved into my house, there was this big, beautiful pine tree kind of between my house and the neighbor's house and came all the way down to the ground, nice and full, all the way down to the ground. Well, one day the neighbor decided to cut all the branches off from six up to about 6 feet. Well, we used to have lots of rabbits, and then suddenly there were no rabbits, just really from that one tree. Since then, I've kind of let stuff throughout my yard grow in. I do not have any mountain laurels or rhododendrons, but I have some goldenrod, and I let the bushes around my house grow up so they're big and bushy and have some low ground cover. But things like planting mountain laurels, rhododendrons, leaving some brush piles, leaving brush and other cover, right? To basically, if I have a conservation soapbox I like to get on, lawns suck. Lawns are biological deserts. Yeah, they're green, but nothing is living in there. Many people coat them with chemicals and other poisons, and nothing lives on them. Let some wildflowers come in, let it grow a little longer, leave the leaves down for butterfly larvae, you know, anything you can do to turn it from this green field of death to something that anything can live in. And even out here, right, um, a square of goldenrod isn't sustaining any species around here. There's goldenrod everywhere, but it does sort of denormalize lawns and normalize letting some of that stuff grow, which is why I do it. So hopefully you learn something about rabbits, but maybe you also don't mow as often. And thank you. And do you have any questions?
No, thank you. Let's,
I'm going to end I will leave the show up in case there's questions from the show. How about that?
Well, no, you can unshare. If I'm going to put a spotlight on you.
Okay. Let me figure out how to unshare.
I can
Cancel.
do that for you.
There we go.
Okay. Well, you're still sharing.
Stop share.
Okay, there we go.
No, stop share.
Right.
There we go.
Now you're in the.
I'm still sharing?
No, you're good. You're good.
Yeah, okay.
And then I'm also going to put, because.
When I turn a light on.
Because Kelly is also on the line. So I'll put a spotlight on her too. Oh, make a co-host so I can review.
See, no, that's terrible. I'm going to actually turn the light on in my room. How about that?
It won't let me put a spotlight on Kelly. But anyway, the research that you were talking about, Kelly Russo is here on the line. So if we have any questions, we can.
Excellent. Yes, she should answer all of those.
And then, oh, look, Steve, our mammal curator here for the Natural History Society is on as well. So he's saying hi to you. Okay, we do have questions. And if you want to ask directly, you can raise your hand. You can put it in the chat box. But let me go back. There are a couple of them over here that were not covered. Is there any difference in temperament? Or what is the temperament?
The only sort of direct behavioral study I found had, I believe, 2 male and 1 female rabbit in an exclosure. And what they really observed was typical rabbit behavior. So if there are, aggressive differences or something between the two, we don't know them. I don't know of any recorded aggressive interactions or anything between them. I mean, it would be really difficult to get because if you can't reliably look at it, right? So you would have to probably have them tagged in advance so you could tell them apart from a distance to see how they would interact. It would be interesting, but
the same thing. Is there a difference in the lifespan of the Eastern versus the Appalachian?
not that I'm aware of, but I didn't. I read mostly on the Appalachian for this, so I would assume the Eastern has a similar lifespan. I can probably look this up in about 2 seconds, but I have a book right here, but not that I'm aware of a difference.
Okay. Is it possible for Easterns to breed with Appalachians? I think you said yes. You were talking about hybridization.
I found one source that I believe was, if I'm remembering correctly, and again, my apologies, being a long day, so I may have the source wrong on this, but I believe it was from one of the DNR sites where the species occurs, mentioning mentioning hybridization as a threat to the species, but I could not find any peer-reviewed scientific papers saying they've detected a hybrid or they found hybridization. And I asked Bill Seton about it, and he has not found it either. So I don't know.
All right. And Tony,
It doesn't seem like they do, but somebody at least thinks they do.
Tony asked, is there could you give the reference for the chromosome studies, or maybe that could be with Kelly? They did some kangaroo rats for the dissertation in the 60s, so very interested in
that. I missed the chromosome study, the
reference from the chromosome studies, I'm assuming for the Appalachian Cottontail.
Versus the New England. I do not have it handy. I can probably. I am not sharing, right?
No, you're not sharing.
Let me. Cottontail. Mammal species. Wrong one. Um, Okay.
Well, you could just send it to me tomorrow and I can send it out.
Okay. Yeah. Um, what? Oh, okay. That's why, because it's not going to come up here. Oh, is this it? Here, what I can do, because I found this, and this is a free link, if I can figure out how to get back. This is Mammalian Species is published by the American Society of Mammalogists, and they're really detailed sort of overviews of the species. Really everything that is known. Um, so I have the link to this. If I can get back to where am I going? I guess I'm going here. And then I will go to chat and I will post that link. So that has it does have an overview of those behavioral studies I talked about a minute ago. That will have the citation for the description in there. Um, all of that. It'll have the chromosome numbers and all of that if you want it. And that's very recent. That is
It looks like it's 2021.
2019. Oh, no, 2008. No, wait. No. It looks like it 2019. It just looks
All
like
right.
took a very long time for it to get.
Has there been any research done with the radio tracking or any plans for that?
There has been Ron Berry, who is my predecessor. So he had my position before me. Did a radio tracking study in Dolly Sods. That's where I mentioned that the radio tracking, they found that they did not enter the open areas.
Yeah. Alan, you have your hand up. Go ahead.
Hi, thanks so much. So I got my standard question, which is when and how did you get interested in this area? Could be going back to as far back in your, childhood, whatever that you want to go. And then my other question, what was your dissertation on for your PhD?
Um, how did I get into this? Science in general is kind of always where I gravitated toward. I had I want to be a marine biologist stage in middle school. There was a brief nuclear engineering fixation early high school. Started undergrad actually looking at chemistry. Um, as I told my classes, sometimes I'm not bad at chemistry, but I do find it extremely boring, which I just kind of drifted into organismal biology mostly. I mean, I grew up hunting and fishing, so being outside, being able to take college classes where we go outside and what's this, what's that, what's this. Um, Getting into small mammals was I originally wanted to get into fish. When I started my master's, there was a better opportunity to work with small mammals than there was with fish, to sort of make a long story short type thing. and then ended up in small mammals, which got me to Panama and Venezuela and Brazil and Alaska, northern Ontario, and these places, which great. Um, Ph.D. dissertation was looking at mahogany logging. So I worked in an area of Brazil that's roughly the size of the state of New York, that's controlled by about 6,000 indigenous people. So, massive, pristine area, not a lot of human disturbance uh, there, but they do sell mahogany trees. And mahogany is valuable enough that, logging operations would build a 700-kilometer road through the jungle to get out, 50 trees. So it's kind of this, we looked at it as sort of this double-edged sword of, here's this disturbance that could have detrimental effects versus here's this relatively minimal disturbance with this huge economic benefit that could actually result in, hey, here's the way we keep the forest standing, because it's certainly better than cattle ranching, which is really kind of what we found. It's isolated impacts, but probably not ecosystem collapse type stuff, which can happen with logging operations elsewhere in the tropics, to try to keep that answer.
Great. Thank you very
Somewhat.
much.
Steve asked, what is known about the phylogenetic relationships of the Appalachian cottontail with other species in the genus? Is it Sylvilagus? I don't know if I said it correctly.
Yeah, that one always trips me up. Sylvilla. Sylvilla. I don't know if I'm going to. If I can pull this up, I have. We can go back. Where am I going? Nope. Here, can I share again?
Yep.
And rodents allow, and now we should see this. So there is the phylogeny of those rabbits we saw earlier. So there's the cottontail in here. The interesting one is actually the volcano rabbit, which is way out here and actually more closely related to those hares. This is down, and I think literally occurs on a single volcano in Mexico. But I guess this is missing our Appalachian, so this isn't all of them. But so the cottontails are in here. So this is your Eastern, your Appalachian, your marsh in there. So I'm sorry, I actually thought that had all of them on there.
All right. Do we have any other questions?
What were those ones at the top of that graphic? The picas? What is that? Pikas?
Pica? Pica are, here. Let's just show a picture of them. Because I have that up, don't I? No, did I close that? Ay. But I can open it easily. get down here to the bottom. Sorry, this is my mammalogy lecture. It's for rodents and for the glaries, so rodents and lagomorphs. It's 111 slides long. There's pica. So if I go back here, go share again. I don't think I get web entire screen. Allow. And then go here. So this is a pica. They're little, they live in alpine areas. This map, you can't see my pointer, I'm sorry. This map, these are fossil finds. So it's the black dots on here are the extant locations, and then in Europe. But they're little gerbil hamster-like things that live in these alpine areas. They have these really big ears, and they can actually close their ears so they can push them kind of over. And they this haymaking behavior, they gather vegetation and stack it into these piles and let it dry out to use in the winter months. And they're really, really vocal. So they sing and make a lot of sound, which is what's in these YouTube videos. And then one of the interesting things with this haymaking behavior is they'll actually cache toxic plants in these caches so that to deter fungal growth. So they'll alternate layers. I got to stop sharing, which I did. They'll alternate layers of toxic plants with the plants that are saving to eat to deter fungal growth.
Fascinating.
And Kelly posted the chromosome difference paper.
And what I really appreciate this is that I wanted to ask you. My question is, are there any plans or thoughts of maybe doing a citizen or community science where we could go out, you have people going out there collecting the scat as well as DNR? More people, more eyes, more hands.
There is not a plan of that currently, but it's actually a pretty good idea. Kelly might be able to answer better how difficult that protocol is. I never did it, so.
Kelly, you want to comment about the study, or?
Yeah, can you repeat the protocol question, or?
She was asking, like, could you set up a citizen science type thing to collect scat? So
yeah, that would be pretty easy to do. Is just let people know ahead of time how to find the tracks, and what the scat looks like, and yeah, I think that would be something you would definitely have citizen scientists do, as long as you make sure they're all wearing gloves and collecting them in the same type of tubes. That should, I think that would be doable. So yeah, it's a good idea.
that might be worth pursuing.
All right. I know that you'll have a lot of people, at least that were on this call, that we could gather up to go out there and do it. So we have any other questions about the Appalachian cottontail? And what I really appreciated also in your presentation was we understanding how science works, and asking the questions and what works and the challenges. So and how we use the information that we gather and make good decisions. So master naturalist would like to do the scat collection. Okay, there you go. Down there. So if we don't have any other,
I will try to remember to talk to Bill tomorrow and see what he thinks.
all right, I think we can get a team of scat collectors out there. And Steve, yes, I agree. Great talk, learned a lot. Thank you so much, Tom, for taking the time after a very long day. We made it longer, but it was well worth it. Thank you, Kelly, for coming on and your research. Congratulations on that. That was great research that you did. And thanks everybody for joining us tonight. Remember, next week for Must Learn Thursday is the wild and wacky, wonderful world of fish reproduction or fish sex. Hope to see you then. Take care. Stay curious, everybody. Bye-bye.
Yeah. Bye. Thank you.